Good evening, everybody. Good afternoon, good morning, wherever you are. I would like to welcome you all to this post-ASH 2022 summary dedicated to multiple myeloma and brought to you by the International Academy for Clinical Hematology. This has become, of course, a tradition every year after ASH, where experts from the ICH would provide you with a summary on the different key topics that were discussed during the usually fantastic ASH meetings. But we are also during the first days of the year 2023, and I'd like first to wish you all a happy new year, a successful and healthy new year. And I always take this opportunity to express on behalf of all the ICH and the hematology community our deeper thoughts and sympathy to patients and families struggling with blood diseases. May your new year be filled with good health. That is really the most important treasure we cherish. So today, for the next 45 minutes or so, I will try to summarize for you the latest advances that I have attended or heard at the last ASH meeting in New Orleans, and it was absolutely a fabulous meeting, and I'm very, very grateful and thankful to the ASH organizing committee for really putting together a fantastic program in a beautiful setting. It has been a true pleasure to be back together in person. As you may guess, multiple myeloma has been also on the podium during ASH. Almost 22% of the abstracts were dedicated actually to multiple myeloma. This is quite incredible that a single disease is really driving so many research. And this is really excellent news for the patient. Obviously, like in any summary, and for the sake of time, I will not be able to go through everything, so I do apologize in advance for those colleagues whose work could not be cited here, but I really acknowledge the hard work, the excellent research performed across the globe in this terrible disease, namely multiple myeloma. These are my disclosures in relation with this topic. Just for the sake of introduction, I don't think I need to spend a lot of time to highlight to this audience the natural history of multiple myeloma. And the story now is starting more and more very early at the level of smoldering multiple myeloma, but also MGUS. I will not go through the fantastic data from the iStop myeloma research coming from Iceland in relation with plasma cell precursors, especially MGUS. But there's one good news to tell everybody, and this is an excellent news for the patients, is that according to data and to the large database and series cohort from Iceland, a bone marrow aspirate is not necessary or mandatory in all cases of MGUS. And that's really very important. I will not also go through what has been presented when it comes to smoldering multiple myeloma, because this is not yet standard of care. I will rather focus on what I personally viewed as important information related to the frontline treatment of both the transplant, but also the non-transplant eligible patient. And the biggest part of this post-ASH summary will be actually dedicated to the relapse setting. And you will see that it is incredibly moving forward when it comes to the relapse setting. Let me start first with the transplant eligible patient. One important piece of information presented during ASH 2022 is about the final analysis of this famous Griffin trial. This is a trial which randomized in a phase 2 fashion the quadruplet DARA VRD versus VRD, daratumumab, lenalidomide, bortezomib, dexamethasone versus the standard of care VRD in induction, followed by transplant, followed by consolidation and maintenance. It has been published. You are well aware of it. What I would like to emphasize here, that with longer follow-up, actually, and looking into the subgroup analysis, progression-free survival by VGPR after induction. So VGPR is really becoming a sort of a threshold, the minimum threshold to be achieved after induction. This is where you have the, I would say, the best results in terms of PFS. But most importantly, we have now the data when it comes to MRD negativity after consolidation. And you would appreciate this really very nice PFS figure at 5 years, showing more than 90% PFS in those patients with MRD negativity after consolidation, further emphasizing the role of MRD negativity. Another piece of information from Griffin here and the subgroup analysis is about the high-risk cytogenetics. And this is in line with the prior data we knew, for instance, from the master trial from Dr. Costa and so on, about this notion of ultra high risk. So it looks like that when you have a quadruplet regimen, whether you have 0 high-risk cytogenetics features or abnormalities or 1, things are doing rather well. It is more complicated when you are in the group of the ultra high risk, more than 2 high-risk cytogenetic abnormalities. Interestingly, and we've been talking a lot about it, the value of gain or amplification of 1q21 is clearly shown here. Of course, these are small numbers, but again, without any statistics or p-values, but it looks like it's rather better to have a quadruplet induction in those 1q values. And that brings me to the issue of those ultra high risk patient. And we are very fortunate now that we have dedicated trials to this group of patients with ultra high risk. We had an update about the Optimum MUC9 trial. This is a trial from the UK. Again, you heard a lot about it. So we had an update about a relatively very this is a very strong, I would say, combination of therapy, almost like a total therapy with DARA VCRD, like a quintuplet, followed by high-dose chemo plus bortezomib, consolidation DARA VRD, consolidation DARA VR, maintenance with DARA-R. So really a very strong treatment package. And you can appreciate that at the end of induction, you have more than 40% MRD negativity, but some patients are going to lose this MRD negativity later after the end of consolidation. So it increases between induction and post-transplant. And this is, I think, an important piece of information in relation with the importance of sustained MRD negativity. And obviously, those MRD positive patients in the ultra high risk who are going to lose their MRD, who are going to lose their MRD positivity, I would say, will do better. And actually, it's not a randomized trial, but the UK colleagues have compared this to their previous trial, the Myeloma 11 trial, and looking into these figures, definitely this new strong package of treatment is rather doing better than the classical standard of care that was used in the UK a few years ago. And this is also in line with another important study fully dedicated to ultra high risk patients. This is the concept trial from Germany presented by Dr. Katja Wiesel, highlighting the value of a quadruplet with isatuximab and KRD, carfilzomib, lenalidomide, dexamethasone. And again, you can see it's a total therapy when it comes to arm A, transplant eligible patient, 6 cycles of isatuximab, KRD, high-dose chemotherapy, auto-transplant, 4 cycles of consolidation, same quadruplet regimen, maintenance, 26 cycles of isatuximab, carfilzomib, lenalidomide. So you would really see. And it looks like, if you look to the median age of the patient, 58 in the transplant eligible, 74 in arm B. And you could give this regimen up to age like 87 in arm B. And you can see the distribution of the high-risk patient. Good news, very high response rate after consolidation. And at the end of consolidation, actually, you have actually more than 70% CR or stringent CR. And this can be achieved with a relatively good safety profile, because when you focus on the adverse events, there's nothing special, I would say. So in summary, clearly quadruplet are here to stay when it comes to the transplant eligible patient and induction. The ultra high risk patient need some dedicated and personalized treatment approaches. What about the elderly, frail, or non-transplant eligible patient? The field is still actually dominated by the results of the Maya trial. As you know, this is really the benchmark trial which has established a new key standard of care in the elderly population using DARA Revdex versus Revdex. And we had an update about the results. You can see the PFS is 52% at 5 years versus 30% for the Revdex. And there is also an overall survival advantage, 67% versus 54% at 5 years. And this is a huge, a 13% difference in overall survival. And actually, while we believe, or people think, that MRD negativity is not so important in the elderly population, still, those patients in the Maya trial who were able to achieve MRD negativity with Dara-Rev-Dex, for instance, you can see here, are roughly around 89% of survival at 5 years. Still 10% more than the Rev-Dex group, even with MRD negativity. So not all MRD negativities are equal. And this is, I think, a very important message. And the other important message, which is, I think, crucial for the from a practical point of view, because from time to time we hear that in the elderly, very old patient, more than 75 or 80 years, maybe you should stick to the doublet combination. This is not true, because if you look to the results in the population over age 75, there is also a clear benefit in these patients. So I believe Dara-Rev-Dex should remain the standard of care, and should be the benchmark for future trials, especially that when it comes to the quality of life, and we know that this is critical for the patient, especially the elderly and frail patient. It looks like that despite that they are receiving continuous therapy, actually there is a preserved and or even improvement in the health-related quality of life items. Well, based on these results, and given the fact that we know now that dexamethasone, high-dose dexamethasone, is really the biggest problem we face in this elderly population, the question is, can we improve over or do better than what we do with Dara-Rev-Dex and actually get rid of dexamethasone? And that was the question asked and presented by Professor Manier from the IFM, a randomized trial comparing lenalidomide, low-dose dexamethasone continuously, to daratumumab plus lenalidomide, but without dexamethasone. You can see the numbers, almost 200 patients in the dexamethasone-free arm, and these are 60% of the patients were above age 80, and this is very important. Of course, this is not the final analysis. Nevertheless, actually, I think the field is ready, maybe, especially when you're using an anti-CD38 monoclonal antibody, to get rid of high-dose dexamethasone. You can appreciate without dexamethasone, actually you have a higher overall response rate compared to the dexamethasone arm. And most importantly, you are going to decrease actually the incidence of complications. And this is, I think, rather good news, and I'll not expand on the MRD issue. So you can see things are becoming quite well established today in the elderly population. The next question is, how can we improve the outcome or the tolerance of the treatment? And this is about the dexamethasone. Probably the next step is about the introduction of the novel agent, like, for instance, bispecific antibodies in earlier lines of therapy. But for the time being, I will move to the treatment of patients with relapsed refractory multiple myeloma. And I have divided this chapter into 3 big sections. One section will be dedicated to I would say, the classical but also novel small molecules. Second chapter will be dedicated to cellular therapies. And last but not least, you will see, we will discuss bispecific antibodies, because if there is one keyword in myeloma from ASH 2022, it is bispecific antibody or T-cell engager. So when it comes to the well-established relapse treatment regimens, we have an update during ASH 2022 about the Aikima trial. And this is an important trial because this is about those patients who are becoming more and more refractory to lenalidomide. You've seen whether in the transplant eligible or non-transplant eligible patient, all of them are receiving lenalidomide continuously. So you need options where you can overcome resistance to lenalidomide or lenalidomide refractoriness. And this is where the combination of an anti-CD38 monoclonal antibody like isatuximab or daratumumab, plus a second generation proteasome inhibitors like carfilzomib can play a role. I'm showing here the oral session, the oral presentation about the Aikima trial, which was dedicated to the outcome of the patient who are in early or late relapse. And this is very important because, first of all, it shows the benefit of combining anti-CD38 and carfilzomib in all situations compared to a doublet, carfilzomib dexamethasone. But the other key message, and I think this is quite logical, but here you have the demonstration, is that when it comes to the late relapse patient, so these are probably less aggressive patients, you have a median PFS with isatuximab, carfilzomib, dexamethasone of almost 43 months. So this is huge, a little bit less than 4 years being achieved in the relapse setting. When it comes to the early relapse, which reflect a more aggressive disease, you can still achieve more than 2 years of PFS. So clearly, an anti-CD38 plus carfilzomib dexamethasone, whenever you can give carfilzomib, for instance, is a very good option. But the story of being refractory to lenalidomide and to IMiDs is going to improve dramatically now with the advent of novel generation IMiDs, and we call them now cell modes. And the two key candidates that are being tested are namely iberdomide and mezigdomide. And I'm showing here the data from the study with mezigdomide, single agent with dexamethasone, oral, 1 milligram, usual 3-week cycle. These are patients who are in relapse refractory setting, more than 3 prior lines of therapy. As you may guess, in the single arm trials, in the really heavily pretreated patient with prior exposure to anti-BCMA therapies. So you would imagine that these are really advanced patients. And you can see here that actually 30% of the patients received prior BCMA therapy. The results are rather good, I would say, especially given that 40% had plasmacytomas. The median number of prior lines was 6. Cytopenia is the key problem when it comes to the use of mezigdomide, and that might explain the infections that were encountered in these patients. Nevertheless, you can still achieve a median duration of response in patients who only achieve PR of 5.1 months, and if you achieve more than VGPR of 9 months, this is very interesting, because I would kindly remind you that when it comes to these very heavily pretreated patients who also received anti-BCMA therapies, having a 9 months after VGPR is really an important, I would say, achievement just by using an oral drug like Meziktomide plus dexamethasone. So very, very encouraging results. And as you may guess, in the next few years, we will see probably Meziktomide moving into the earlier lines of therapies. One very exciting and attractive, I would say, study I have noticed is this one, which is testing for the first time an innate immunity engager in multiple myeloma, namely Modakaphas. So this Modakaphas alpha selectively binds to a unique epitope on CD38. And this is different to the binding epitope of the classical anti-CD38 monoclonal antibodies we know, like daratumumab and isatuximab. And again, single arm, dose finding study with an escalation phase and expansion phase. The median number of prior lines of therapy here, we are adding 1 line. It's 7, from 3 to 19 or 3 to 16. And 85 to 93% of the patients were already refractory to an anti-CD38. Despite such advanced disease, you have an overall response rate around 43%. Follow-up is relatively short, 5.3 months, but not so bad. The median duration of response, 12.5 months, and the median PFS, almost 6 months. So very attractive, in my opinion, when you have already failed so many options, 7 lines of prior therapies as a median. And of course, if you look into patient with an earlier disease stage, like patient who were not exposed to an anti-BCMA agent, actually your response rate is roughly 60%. So especially you can achieve all this with an excellent safety profile. I was quite impressed by the safety of this agent, and I'm sure we'll hear a lot about it in the next few years. You can see here in the indicated in the fixed dose equivalents of 1.5 and 3 milligram per kilo single agent cohort, they are now being investigated as part of the extension study, and I think we'll hear about it next year. So this was the story of the small molecules. But of course, when it comes to relapse refractory multiple myeloma, cellular therapies are all over the place. I think we're very fortunate having already ide-cel, cilta-cel being approved and available in a few places. It's always unfortunate that not all patients in need are able to access them, and we really, I really would like to advocate on behalf of the whole community that we need to facilitate availability, accessibility, but also affordability of these innovations. And this is why I think there is a lot of research on trying to refine, to optimize, to improve the CAR T cell. And one example is this dual BCMA and CD19 targeting, which has been used here as first-line therapy in the high-risk transplant-eligible, newly diagnosed multiple myeloma. This is the GC012F product construct. And you can see the design of the trial. And why is this interesting? Well, first, apparently, it is generated fast, very quickly, not several weeks, but also it has this dual target. Of course, we may question whether CD19 is an appropriate target or not, but I kindly remind you that the first case report of CAR T cells in multiple myeloma in the New England Journal of Medicine was actually a CAR T cell from UPenn against CD19. You can see here, what is really interesting is the very high response rate. Again, the follow-up is relatively short, but we can criticize the early MRD assessment, but I think these numbers are very attractive, very appealing, and maybe they are setting the stage to bring CAR T cells very early in the management of the ultra high-risk patient, especially that when you consider, for instance, this construct, when it comes to side effects like CRS, cytokine release syndrome, or ICANS, the neurotoxicities, it looks like that only grade 1 and 2 CRS or ICANS, CRS were observed, and they resolved actually rather quickly. When it comes to other side effects, well, these are the usual, I would say, hematological toxicities, but I think we in hematology are well familiar with this. When it comes to CAR T cells, I have also selected this cohort 2A from one of the KARMA studies presented by Dr. Usmani. And this is about the patient who are in early relapse after auto transplant, because I think an early relapse after auto transplant clearly defines a high-risk population. The high-risk definition, in my opinion, is not only a baseline when it comes to cytogenetics, but also it's a dynamic process, not only with MRD, but also, for instance, early relapse. And you can see the definition here. It's less than 18 months from initiation of therapy. And then one can envision that you would like to give these high-risk patients an effective therapy like CAR T cells, and this is here, ID cell. I will not go through the characteristics, although I'd like to bring your attention that it goes up to age 77. So interestingly, this is the concept, in my opinion, that you can be CAR T cell eligible and not necessarily transplant eligible. So this is something our lymphoma colleagues are working on, but I think this is very important. And you can see here the overall response rate, almost 84%, so very interesting. And when it comes to MRD negativity, again, we are in line with what you would expect from a CAR T cell construct like ID cell. The median duration of response in all responding patients is around 16 months. These are, by definition, in my opinion, very high-risk patients. And the median duration of response in patients who achieve more than CR is 2 years. So clearly, very attractive results, which are really setting the stage to probably push to use CAR T cells and cellular therapies and immune therapies in general, because we'll talk about the bispecific antibodies in the early relapse population, and you can see here the side effects, but also the PFS figures and OS, and median PFS is 11.4 months. This is very nice. There is also another now construct with CAR T cell being presented at ASH by Dr. Costa. You can see here the design of, sorry, yeah, here I'm back. So you can see here the study design of this fully humanized construct. And again, these are patients with ultra high risk features, 38.5% of extramedullary plasmacytoma, high risk cytogenetics, median of 5 lines of prior therapy, 91% were triple class refractory. And I think this is really important, such type of construct, because these cells may probably be more effective in the population. And we can see here, for instance, an overall response rate of 100% and 71% of CR, of stringent CR. So really, this is allowing to upgrade, I would say, the level of response, and you can achieve this without an increased risk of CRS or neurotoxicities. But that was the cellular therapy. These were the cellular therapy products targeting BCMA, and we know BCMA is really the champion here. But what has been really very nice during the last ASH, especially that now a lot more and more patients will receive BCMA targeted agent, is to have cellular therapies targeting others than BCMA. And this is, for instance, the construct targeting GPRC5D. And we'll talk later about GPRC5D in the bispecific section with talquetamab, which is leading to really some amazing results. But here you have a CAR T cell construct, and this is different to the one that was published in the New England Journal of Medicine a few weeks before ASH, by the way. This is another one. Again, you can see the age, it goes up to 80. These are slightly less advanced patients because the median of prior lines of therapy was 4, not 5 or 6. But again, you have 45% extramedullary disease and roughly 50% of the patient with high risk. What's really interesting is that you have 40% of the patient, 13 patients out of 33, who received a prior BCMA-directed CAR T cell therapy. And of course, it has failed, this treatment. So here, well, the idea is say, okay, it didn't work when I targeted BCMA. Why I don't try another target, GPRC5D? Bingo, it works. It works very well. And you can see at the dose level of 150 million, for instance, you have 100% response rate. And actually, the overall response rate in patients with and without experience of BCMA targeting therapy is really excellent. Obviously, slightly lower when you are in a more advanced disease. I'm very passionate and excited about these results, but again, we need to be humble because the numbers are always small, like 9, 10, etc. But I think there's no smoke without fire, or fire without smoke. This is what we say in France. So there is a clear signal here about the fact that targeting another tumor antigen can be important and can actually salvage those patients who received and did not respond or relapse after an anti-BCMA agent. The side effects are again relatively in line with what one would have expected, especially when it comes to ICANS type neurotoxicity, very low, and there are no dose, I would say, significant limiting toxicities, because this is about more, I would say, hematologic toxicities, which we know very well in the field of multiple myeloma and hematology in general. Last but not least, when it comes to the treatment of relapse refractory multiple myeloma, is the story of bispecific antibodies. Of course, we already know, and we already heard a lot in the last 3-4 years about bispecific antibodies. And we're very lucky now having one bispecific antibody, namely teclistamab, being approved and increasingly used. And this is targeting BCMA. But the good news is that we have now really more and more effective and other alternative. And this is always important to have different alternatives. Here you have, for instance, the results of elranatamab. So elranatamab is another bispecific antibody T-cell engager targeting BCMA. And we had here the results of one of the majestic early phase trials presented by Dr. Nupuraj, 55 patients, median prior lines of therapy 5, 98% were receiving prior anti-CD38 antibodies, monoclonal antibodies, and 91% were triple class refractory. Initially, I think the fear of the community when it comes to bispecific antibodies and T-cell engagers was about CRS and maybe neurotoxicity. Well, actually, it doesn't look like that this is the key problem we are facing with bispecific antibody. And actually, the setup priming dose, as it has been done here with elranatamab, has allowed to successfully mitigate the CRS. And you can see here the responses. We have a decent follow-up here, 12 months. The median duration of response, 17 months, and you can see very nicely sustained responses. And now there are more and more MRD negative patients. And you can see these are really highly advanced patients and a high-risk patient. So I believe elranatamab is going to be another key player in the family of the anti-BCMA bispecific antibodies. But there is more to come, and we have to learn how to pronounce all the names of all of these new agents. And there is a new member into the bispecific antibodies family, namely alnuctamab. The development of alnuctamab is slightly less advanced compared to teclistamab, which is approved, elranatamab, which is quite advanced, but still it is an attractive one. This is a phase 1 setup dosing of alnuctamab. Again, a lot of high-risk patients. I will not go through the data. I simply wanted to mention that there will be a lot of great options, and all of these new bispecific agent are going, I think, to make it, and we have to learn about the right combinations and where to position each of these drugs, because of course the space is relatively over crowded, but I think it's always a good problem to have many agents. And you can see here that with subcutaneous alnuctamab, you can achieve, the bar is quite high, 65% overall response rate, and the median duration of response is not yet reached. And 90% of responses are ongoing with a lot of MRD negativity. Well, if all of these bispecific antibodies are doing great as single agent, well, why not combining them with other effective agent and then brings them a little bit earlier in the course of the disease? And interestingly, and some people thought that, oh, you cannot combine antibodies together. Well, you have here this Majestic 3 trial design using darasubcute single dose lenalidomide plus teclistamab. Very interesting. Remember, dara-lendex, standard of care in the elderly population. And here you're adding to this combination, not in the frontline setting, in the relapse setting, but early relapse, because the median prior lines of therapy was 2. You're adding teclistamab, which is already approved as a single agent. And well, the median follow-up is, in my opinion, still relatively short, because we are talking about early relapse, median of 2 lines. But you can see that, first of all, the median time to the first response is very quick, 1 month. And that's important because it allows to decrease the symptoms of the patient. Very high response rate. And it's a good start because the responses are deepening over time, and the median duration of response is not yet reached. But you can see there is a trend for a sort of a sustained progression-free. And while one would be worried about such combination in terms of safety, actually, when it comes to the CRS, grade 1 and 2 only, so not really a big, I would say, matter of concern. I would like to point out, however, the issue of infections and opportunistic infections. And there is a biological rationale for this. Obviously, it hasn't been studied here, but in general, and what has been shown in the different studies with bispecific antibodies, and probably because of the phenomenon of T-cell exhaustion. And we know very well the importance of T-cells, not only in anti-tumor responses, but also in anti-infectious responses. Definitely, there is an important concern about the issue of recurrent infections or opportunistic infections or viral reactivations. It is not very easy to really have a clear picture who is doing what here, because we're talking about relapse patient who received a lot of dexamethasone, so they're highly immunosuppressed. And in addition, you are inducing some T-cell exhaustion with the bispecific. So I think we need to pay attention, and there is some work to be done here in order to mitigate the issue of opportunistic infections. Similar to the CAR T cells, actually, assuming majority of the patient will be able to receive an anti-BCMA bispecific antibody, one may wish to figure out another target. And that brings me to GPRC5D, the one I have shown you data with a CAR T cell construct. And this is about talquetamab and the monumental one study. And that was really a lovely study and coincidence, because the paper was released and published in the New England Journal of Medicine simultaneously to the presentation by Dr. Ajay Shari. And you can see here with talquetamab, which is a T-cell engager targeting GPRC5D, you can appreciate that patients have received 5 lines of prior therapy. They have received anti-CD38 and they were refractory, 93%, triple class refractory, but also they have received some prior T-cell redirecting therapies. So if you look to the overall response rate today with talquetamab in heavily pretreated patients, the bar is very high, more than 70%, almost 75% with the 0.4 milligram per kilo subcute. If you look to the patient who already received a T-cell redirecting therapy, you're still around 63%, probably lower than 73%, but I'm not trying to do any statistic there. But this looks very promising, because it means probably you can come back with another T-cell engager targeting another tumor antigen and then mobilize, I would say, the immune system against multiple myeloma. And the follow-up is quite decent, a year, median follow-up almost 1 year, and the median duration of response is almost 13 months. I think this is really, again, another benchmark when it comes to the relapsed refractory setting, because it works also in those patients who received prior CAR T cells or prior bispecific antibodies directed against BCMA. You can see here the median duration of responses, and the responses are looking quite durable. CRS or ICANS, in my opinion, still quite manageable. Very small numbers of grade 3, 2 cases at 0.4, another 2 cases of grade 3 at 0.8. But the issue, as I mentioned, for the anti-BCMA T-cell engagers or bispecific, the issue is about the infections. And I will not go through the details of the opportunistic infections, but clearly this is happening, and we need to do prevention, to monitor these patients in an appropriate manner to avoid these infections. There's more yet to come, and you have this other bispecific antibody, the Regeneron 6234, the Forimtamig, and again, a dose finding study. And you're going to see always, you can give up to age 80 plus these bispecific antibodies, despite the issue of infections. Interestingly, here with this study, early study, you have both IV arm and subcute arm. You have 22% of the patients who received prior BCMA-directed therapy. And again, the key message is that this other, I would say, a bispecific antibody leads to a relatively high response rate. So we're talking now, looks like the benchmark is 65 to 70%, even in the heavily pre-treated patient. And the median duration of response is around 10, 11, 12 months. So I don't believe today one can really distinguish between all of these bispecific antibodies. We need to work a little bit more, trying to refine and to see how we can distinguish, or how we can differentiate and use in an optimal manner these bispecific antibodies. Last but not least, we have also bispecific antibodies targeting another antigen, namely FcRH5. And this is about, for instance, this study. Again, I will not go through the design of the study, but interestingly, if you read carefully, the key inclusion criteria is the use that was allowed of prior CAR T cells, antibody drug conjugates, but also other bispecific antibodies. Again, I would like to emphasize the median number of prior lines of therapies, but also triple class refractoriness. And this cevostamab agent, which actually has been given for a fixed duration. And this is interesting because from a patient perspective, from a health economic perspective, it is very attractive to try to develop treatments for a fixed duration. And for those patients who are able to complete, for instance, 17 cycles, and you can see the study design here, up to 17 cycles. Well, median follow-up roughly 10 months, but I think this plot is really very nice, showing some durable responses. Again, maybe the fixed duration may allow to mitigate the issue of T-cell exhaustion, but I think this is really very, very attractive. And definitely by using preemptive or early tocilizumab, you can definitely reduce the CRS rate with no negative impact on the anti-myeloma activity. So in summary, well, I would need another hour, I would say, to give you a summary of what's ongoing. So I thought that if we really consider the latest advances in multiple myeloma from ASH, this is definitely the immune therapy revolution. So with this, I'd like to thank you all for your attention. And I can see already dozens and dozens of questions. So there are questions from colleagues asking me if we can share these slides. Well, it depends. Please write to me and we'll figure it out. We have a question about whether we consider gain and amplification 1Q as a bad risk factor. Short answer is yes. This is a poor prognosis risk factor, but obviously there is a debate about the number of copies and so on. So I think we need some time, but I think the message I conveyed was about that. The quadruplet agents are rather a good candidate, and whenever possible should be delivered to these patients. There are a lot of questions about MRD, and it looks like everybody is convinced about MRD. One question is whether, again, if you achieve MRD negativity after induction, should we perform auto transplant? My practical answer is still yes. maybe in the future, if we are able to demonstrate the sustainability of MRD negativity, and I think the master trial is a good example of trying to guide therapy based on MRD negativity. But today, I think, at least in a European perspective, while we're very happy to achieve MRD negativity after induction, I believe we should continue to give high-dose chemotherapy for those who are transplant eligible. We do have a lot of questions about, it looks like these are like colleagues are asking about specific cases, in case of renal failure, in case of CNS localization. There's an interesting question here about my opinion about the role of stem cell boost. Well, this is interesting. So I don't know the context of the question, but for instance, it is more and more frequent to see after CAR T cells in multiple myeloma, for instance, to see some long-lasting cytopenia. And we know this also from the lymphoma field. And obviously, if you have already stem cells stored and available, this can be a good option to give after, I would say, CAR T cells for cytopenia. Well, we have a few questions about what to use if you have early progression after CAR T cells. Well, today, I think, and there are lots of questions about the right sequence of treatment. I will not be able, guys, to answer today the question of the sequence of how to sequence all of the treatment of all of these treatments. This is a $1 billion question, so it's going to be impossible to answer this. But definitely, I prefer CAR T cells and then bispecific. Clearly, if you have targeted BCMA, I think now with the availability of other targets, and by the way, talquetamab has been filed for approval by FDA at the level of FDA, but also at EMA 2 days ago, I think, on the 3rd of January. So having another target and trying to give a drug, an agent, which can target something different like GPRC5D is definitely today, in my opinion, the optimal approach. And again, of course, it is about the availability of the agent. Well, there are questions about peripheral neuropathy with bispecific antibodies. I would say from what we have seen with teclistamab, elranatamab, alnuctamab, and the Regeneron one, and the other bispecific, I don't see there a big signal when it comes to peripheral neuropathy. But also, again, these are not randomized trials. Usually the numbers are relatively small. But most importantly, guys, please don't forget that the patient who are receiving these bispecific antibodies, they have usually received bortezomib, for instance. They have, some of them received thalidomide, and they have already a pre-existing peripheral neuropathy. So you need to have a careful monitoring of these patients. And one cannot today, I would say, blame or say, well, peripheral neuropathy is a big problem with bispecific antibody. We have, again, a very nice question from Professor Dominique Braun in Belgium. And this is again about, well, she wrote, given the, and I'd like to say hello to Dominique. She's really a master in the field and has done a great job in hematology over the last 30, 35 years. Taking into account the excellent PFS of older patients in MRD negativity after DARA VRD, could we postpone auto? Well, that is now the question, I think, that will be answered by some of the ongoing trials, especially the IFM trial, the MIDAS trial, which is, by the way, open also in Belgium. I have a few questions that I didn't mention, the monitoring of the disease with the new techniques like mass spectrometry and circulating DNA. I do acknowledge that these are important topics, but already in my introduction, I highlighted that unfortunately, for the sake of time, I won't be able to go through everything. So I focused on the therapeutic aspect. But I fully agree with you, Dr. Shen, that circulating tumor cells and tumor DNA, but also using maybe mass spec to analyze MRD negativity is going to be something important for the future. There is a very interesting question, very important. I don't know the answer, unfortunately, from Dr. Hira Mayan in Canada, saying, well, in the KarMMa-2 trial, where, you know, these are earlier relapse, would we have expected a better median PFS? The answer, I would say maybe. But the other way to see things is that, well, the efficacy of immune therapy would be roughly the same when it comes to CAR T cells, for instance, irrespective of your disease stage. This is pure speculation, but I agree this is an important question to ask, because we all believe that if we use immune therapy earlier in the disease setting, we would expect better results. The only concern I always have is that we do not have actually randomized data all the time to have these final answers. But there's always like this competition between going fast with a single arm and making a drug available to those patients who are in need, and on the other hand, doing the randomized trial. So I know there are pros and cons, and there's no single answer or black and white answer to all of this. We have a few questions about whether there are differences between results of DARA-based combination versus cetuximab-based combinations. I think the most important part of the story is that, in my opinion, more and more, I mean, the majority of the patients should receive anti-CD38 monoclonal antibodies. They are not the same antibodies, daratumumab or cetuximab, but definitely they have to receive anti-CD38. I think the goal is to make anti-CD38 monoclonal antibodies to all patients with multiple myeloma, because today, proteasome inhibitors, bortezomib being generic, IMiDs, lenalidomide is a generic, and having an anti-CD38, you have already, and I'm talking here when it comes to the low and middle income countries, you have already here 3 pillars of a successful and highly effective combination of myeloma therapy. And I'll take one question, one last question, which was asked actually in French, whether for those countries with low resources, whether it is better to invest to implement cellular therapy infrastructure or just use bispecific antibodies. I don't have the answer, my friend. it's always good to have access to all options. Bispecific antibodies are off the shelf, easily available, and I think when we will have all the recommendations, how to use them, how to mitigate the side effects, the opportunistic infections, definitely they will be quickly available compared to CAR T cells. But I think cellular therapy has clearly a huge added value for patients, because for the time being, it is a single shot treatment. And we should never forget the patient perspective when it comes to designing and running all of these trials. And that will be my last word. I hope you have enjoyed this post-ASH22 webinar by the IACH. I would like to thank you all for your loyalty. It has been quite incredible, the number of attendees today. I think we've broken a record number of attendees. I don't have the exact figure, but the technical team is giving me a signal about this. We went a little bit over time. We usually try to stick to 45, 50 minutes, but this is really so exciting. So thank you so much for watching. Thank you for following. And you will hear more about the post-ASH summaries by the ICH. We're planning a summary for CAR T cells in general, a summary related to stem cell transplantation by Professor Nagler. We'll have a summary for MDS, MPN. So a lot is yet to come from the ICH. Stay tuned. Happy New Year. Best wishes for a healthy and successful New Year. And wherever you are, please stay safe and keep well. Thank you very much.